Okay, so we'll begin our presentation. So for those who are watching the live stream, welcome. And this is our first Rutgers Camden MARC bioinformatics training presentation. So before I forget, I want to acknowledge the OARC, Office of Academic Research Computing, really collaborated with us, and Dr. Janet Chang, who instructed our student the coding. I appreciate that, your help. And Tom Skipper also helped us, our student, just properly setting up for accessing the high HEPC. So we recently purchased a node of MRL, and it was the intention to teach our student bioinformatics skill set. So this week, whole week, even before semester begins, they were here learning coding. And first 2 days, they learned how to do sequence variance analysis using human genome. So they finished analyzing genetic disorder, and they have a presentation on that. And then they were challenged to come up with a research project using COVID-19 sequence. And well, it's not the proper name. COVID-19 is a disease, right? So the coronavirus sequence, they can download from public database. And the challenge is that they have to come up with a hypothesis-driven research question. And then using the skill set of sequence variance analysis and have some new discoveries. So we have a rising junior student, rising senior student, and this is actually a very challenging question, even grad student, but so I didn't know what to expect. So for the last 2 days, they work as a team, junior cohort and senior cohort, and you will hear about what they found out. So I just want to emphasize that I'm very proud what they've done. Excited to show you what they have accomplished in 2 days. So the question and process conclusion is all by them. So you did a good job. So now, senior cohort, if you're ready.
So we're the senior cohort. My name's Anthony Monte Carlo.
My name is Tiffany Scoher.
I'm Juliana Jimenez.
I'm Sienna Cachado.
And this is our presentation on our analysis that we performed for the SARS-CoV-2 B117 variant, better known as the alpha variant, and its trends and mutation.
So, as we all may know, SARS-CoV-2 is a coronavirus that causes respiratory illness known as COVID-19. COVID-19 was first detected in Wuhan, China back in late 2019 and gradually made its way around the world, causing the global pandemic that we had. And since then, different variants of SARS-CoV-2 have arose due to these mutations in the virus's genome. The first variant that was detected was in the United Kingdom in winter of 2020, known as the Alpha. And since then, we've also had the Delta, Gamma, and Beta variants that have risen. And the Alpha variant is known to have an increased transmission rate of nearly 50% and potentially attributes to increased fatality rates. So, since COVID or SARS-CoV-2 and the different mutations have skyrocketed, and the severity of this public health situation has become more increasingly concerning, we wanted to focus our research on how fast or how much does the variant, alpha variant in this case, mutate over a short period of time.
So for our project, we focused on the data from UK because we know that the alpha variant was first detected in the UK. So here's the graph that shows the timeline on the x-axis, and on the y-axis we can see the number of COVID cases in the UK. So we can see that the COVID first appeared in March, and then after that, we know that in the late winter, in November or September, the Alpha variant was first discovered in the UK. And right after the detection of the Alpha variant, we see a spike in the number of COVID cases. So for the purpose of our study, we will focus on these 4 months, November of 2020, December, January, and February.
So the question that we are trying to answer in our project is if the rate of mutation in SARS-CoV-2 changed during these 4 months, and if it did, if there was a correlation between this change and the infection rate. So we hypothesized that the rate of mutation of the SARS-CoV-2 virus will be directly proportional to the increase in cases.
So as Dr. Lee has mentioned, we recently acquired our own personal node on the Amarillo High Computing Cluster. And so here we outline the way that we went about collecting our data, processing it, and then drawing our conclusions. We first searched the NCBI database for a number of sequence reads that came from the UK, were sequenced between the months of November 2020 and February 2021. Got the accession numbers for those samples, and then pulled them directly to our cluster using NCBI provided tools like FasterRQ. The first step in actually processing the data was to quality control it using FastQC, then followed by, after verifying that our samples were able to be used, trimming and mapping those samples using the GATK pipeline. Following that step, we then converted our resulting files to a format that can actually be read by other programs, from SAM to BAM, and then sorting and indexing those files. And then again, another quality control step. After that, we then search our samples for variants using the variant calling haplotype caller feature in GATK's pipeline. And then following that, we then annotate our samples using the SNP-F tool and then generate summary files. Because of the length of time that this process takes, we had to limit the number of our samples, and so we were mainly focused on analyzing 3 samples for November 2020 and February 2021, and then we only analyzed 1 sample for December 2020 and January 2021.
So after we gathered all our data, we were able to put it into a bar graph format. And as Anthony mentioned, we only are viewing or did statistical analyses on November and February, because November being the earliest or pre-spike period and February being the aftermath of the spike in 2020 to 2021. So this was the total number of mutations by month. So these mutations include SNPs, insertions, and deletions. And since we are unable to currently determine whether this is there's normality or no normality, we just use both Welsh and Mann-Whitney statistical tests. So for the Welsh one, we obtained a p-value of 0.03, showing that there is a significantly statistical difference between the number of mutations in November and February. But on the Mann-Whitney one, we had a p-value of 0.10, which shows there was no statistical significant difference between the number of mutations. But we do see an increase in mutations on the genome after or during the spike period of COVID cases.
So here we show the rate of mutations broken down by month, and the mutational rate of our genome is calculated by the total nucleotide length of the genome divided by the total number of mutations found by our programs. So a higher ratio would indicate a better conservation of the genetic information in our virus, whereas a lower mutational rate or a lower ratio would indicate a higher variation. And so as we show on our graph, we have a higher mutational rate, meaning a higher ratio, and more conservation in our genome in the month of November versus the month of February. And so again, we ran both statistical tests, whereas with the Welsh test, we got a return p-value of 0.047, whereas with the Mann-Whitney test, we have a return p-value of 0.1.
So we then also compared two different types of mutations between the month of November and February. The orange bar here shows the missense mutations, and the blue bar shows the silent mutations. So for the missense mutations, when we compared the month of November and February, we found out that there was a significant difference in the case of Welch test. However, when we ran the Mann-Whitney test, there was no significant difference. In the case of silent mutations, no significant difference in the number of silent mutations was observed between these 2 months, the month of November and February. So looking at the trend, we can see that these 2 months, the first 2 months have lower number of missense mutations, and the next 2 months, January and February month, have higher number of missense mutations. However, the silent mutations do not increase.
So next we read a graph on the number of mutations that were located in different areas of the genome per month. So for this, we just focused on the exons and the introns. So the general trend of this graph shows that there are more mutations located in the exons compared to the introns. And also it shows that in November there were less mutations in the exons than after the peak in January and February. So in November and February, we also did the statistical test on them using the Welch and Mann-Whitney test. So for the exons, we found that between November and February, using the Welch test, the p-value was 0.028, meaning that it was statistically significantly different. So there were more mutations in the exons in February than compared to November. However, when we used the Mann-Whitney test, we found that there was a p-value of 0.1, meaning that it was not a significant difference. Also, in the introns, we did the same 2 tests, and the p-values came out to be 0.1 for the Welch test and 0.4 for the Mann-Whitney test, meaning that neither of them showed statistical significance.
So one of the conclusions we've drawn from these graphs was that there was a trend in the missense, an increase in missense mutations in January and February, which does explain the increased infection rate in January and February.
Well, since we know that most of the mutations were on the exons instead of introns, and exons are transcribed into mRNA, which is then translated into protein, we can predict that this higher rate of mutation in the exon can change the functionality of proteins.
We also concluded that our data suggested that most of the mutations being in the exons, that the virus was feeling some evolutionary pressure to mutate in a beneficial way that would help spread the virus and make infection more beneficial.
So for our future plans, we will analyze 3 or more SARS-CoV-2 sequence samples for each of the 4 months so that we can perform statistical analyses on all of the months and draw proper conclusions.
These are our references. And we would like to thank Dr. Lee for creating this workshop, bioinformatics workshop. Dr. Janet Cheng for teaching us how to code in such a short period of time. Tom Skipper for any technical issues with Emerald. MARC U-Star program for funding us. And Dr. Nathan Free for being co-director and supporting the MARC program. And Rutgers University Camden for being our school.
Okay, so there's a question from Dr. Geneva. Let me just read it for you. Can you explain again the way you calculated mutation rate?
Yes, I get it. Do you want to go back to that? So the way that SNP-F had calculated the mutational rate was it took the total number of nucleotide changes, whether it be SNPs, insertion, deletions. Those were sort of the 3 main changes that we had seen in our data. It takes the total number of those changes and uses it to divide the total nucleotide length of our genome. And so it comes up with a ratio of, I think it was maybe 16, or rather maybe like one change per 1,300 bases,
Yeah.
give or take, for November data, whereas February it was one change per 800 bases or so.
Yeah, I think that I was confused by that too. I think couldn't it be better to be called conservation rate? So mutation
Yeah.
rate, the way you say here, is different than the next slide here. So you said the number of mutate is kind of confusing.
Yeah, I agree.
Yeah, so this is more a proper way of saying mutation rate. And previous one I would call conservation rate. If there's such a thing, I think the mutation rate is the other way around.
Yeah, we just couldn't come up with a better title.
Okay.
Because yeah.
Yeah, so there's another question. Did you use the same number of samples per month?
So for November and February, we used 3 samples for each month. For December and January, we only used 1, and so we sort of disregarded those months in our statistical analysis.
So he's saying, I'm asking because if you have more samples later in the outbreak, you would expect there to be more mutations in those additional samples.
We didn't have more samples. So the number of samples for this month and the month of February were same.
Good answer. Any other questions? So what was most challenging aspect as you do this project for you?
For me, it was coding and trying to understand what mistake is resulting in the errors.
I also thought coming up with a question was also challenging and finding out what question we could do in the short amount of time that we had.
I would say understanding the data, because I have yet to take a genetics course, so some of the stuff was like brand new information to me. So trying to inform yourself enough to know what you're talking about in such a short period of time was definitely like a lot of pressure.
Yeah, I think also just making sure that we all sort of understand each step, I think is also a little bit tricky, because we could definitely sort of get lost in the actual coding process a little bit. And then also I think just making sure that we have enough time and sort of guessing how long it might take to run each of these, because it could vary a little bit. That also is a little bit challenging.
Yeah, I commend you that in 2 days you come up with an authentic question and then have a data for it. So I am very happy about what you have done. Okay, good job. Good. Bring your book.
Do you guys need this at all?
Yeah.
Okay.
Okay. Hello, my name is Dier Kabaski.
I'm John Cresswell.
I'm Sishoya Dugan.
I'm Teresa Sorio. We are the junior cohort of the MARC program here at Rutgers Camden, and today we're presenting our findings after a week of bioinformatics training. And hello, my name is Dier Kabaski.
Yeah, you can mute that.
We are the junior cohort of week at identifying common mutations in the SARS-CoV-2 variants B117, which is the alpha variant, and B16172, which is the delta variant. Coronavirus is a disease caused by the virus SARS-CoV-2, which first emerged in October 2019. COVID-19's ability to transmit has led to a global pandemic that is still going on today. And the virus SARS-CoV-2 has undergone several mutations and created variants of the original virus that have different symptoms and different rates of mortality, morbidity, and prevalence than the original virus.
So based on this information, our goal of the study is to use variant analysis techniques to identify the location of these mutations and compare and analyze the similarities between the alpha and delta variant. So based upon this, our hypothesis is that we hypothesize that alpha and delta variants will have one of the same mutations on the spike gene of the SARS-CoV-2. So we'll be going more into detail about this gene.
So the B117 or alpha variant was first reported from the United Kingdom in September 2020. This variant is different than the original because of a 6 nucleotide deletion mutation on the S gene, and this deletion resulted in spike gene target failure, or SGTF, which prevents the amplification of the spike gene in the disease. The Delta variant was first reported in India in December 2020, but it first appeared in the United States earlier this year in March 2021. It is now one of the most dominant SARS-CoV-2 strains, and current studies indicate that this variant has higher morbidity and mortality than the original. This variant also has mutations on the spike protein, and based on this information, we think that the Delta and Alpha variant will have one of the same mutations.
So to give a little more background on the S gene itself, it's very important for receptor recognition as well as cell membrane fusion within SARS-CoV-2. So mutations in the S gene have caused the gene itself to not be expressed within the alpha and delta variant. When this gene is not expressed, it's called spike gene target failure, also known as SGTF. Evidence indicates that COVID-19 of the cases of COVID-19 within the alpha variant with SGTF have higher rates of mortality and lower estimated rates of survival than cases of the original strain. In this graph here, we see that there is a much lower rate of estimated survival in cases that had that SGTF gene over time in comparison to cases without the SGTF.
Based on this information, we will be comparing the genome of the alpha variant to the delta variant to identify any similarities and hopefully to pinpoint the location of the mutation. So here is the exact mutation that we are hoping to identify. It's the D614G mutation, and it's on the spike gene, and both the alpha and the delta have it, and we're hoping to confirm that and to identify that on both of those genomes.
So open source sharing of viral genome sequences have allowed researchers to track emerging SARS-CoV variants. In this study, we compared genomic sequences of SARS-CoV-2 variants, which were alpha and delta, to investigate the common mutations that are associated with the mortality rates that we discussed previously. So these samples were specifically collected from the NCBI SRA site for genomic sequences. After the processes of collecting said data, we went through different GATK pipeline methods The first being quality assessment, where we use software's fast QC as well as Trimmatic in order to filter the data to make sure that it was of standard quality. Following that, we used the BWA software to map the data and do realignments.
After that, we did the variant identification as well as the annotation. This is where we used the GATK environment, and this is where we categorized all the variants in order to produce our report, and that's where we used visualization. So that's where we received a report and a DNA sequence as well between the three samples. So you can see over here, this is the location of the DNA sequence between the three samples with an N of 1, where we used the location 23,403 base pair. And in the right image, the top one is the delta sample, and the bottom one is the alpha sample, where you can see that they're all aligned in the same position, showing that there is a mutation in this base pair as well as in the original. And over here, you may see that although this shows that there's a similarity, where can you differ the details between these three samples? And then that's where we used this report, and we did a variation count between these three regions, the downstream, exon, and upstream, where we can see that there is a difference in variation between three regions, where in the upstream, the delta has the most variation in comparison to alpha and baseline or original. And since the S gene is located in the upstream, this is our main focus. And there was also an N of 1, so we could not perform a significant difference. Since this workshop was for 2 days, we were not able to produce more than 2 samples. And yeah.
So in conclusion, based on all information that we gathered, we conclude at base pair 23403, there is a missense mutation in both the alpha and delta variants. So based upon this, the 3 samples, the location was taking place in the same region. So there is a difference in the amount of mutations taking place, which provides a potential location for looking into future mutations for future variants. So because of this, we can say that pinpointing the gut variant proved to be very difficult, but expert research confirmed our hypothesis, in which Derica will go over, I think, in the next slide.
So we cannot confirm that the common mutation between the alpha and delta variant is the D614 mutation because of the fact that many articles talking about the location of the D614G mutation did not disclose the exact location. We can only assume, based on the fact that from the alpha variant to the delta variant, the only mutation that is consistent is the D614G mutation. We can only assume that the mutation that we saw based on our IGTV image was that mutation. So hopefully in the future there'll be more openness in other reports so that when doing this type of method, people can find the exact location and be able to identify which mutation they're looking at on the genome.
So just like was mentioned earlier, this was a week-long project with 2 days of work, but with more time, we would compare even more variants and use a higher sample size rather than one. And the same method we implemented can be used in the future to test newer variants and understand how the virus is mutating and if new variants are mutating from older variants. And also we can use this specific location to test for these variants. And this technique can also be used with other genes to identify similar mutations between variants.
And we'd like to extend our thanks to Dr. Lee for helping us throughout this week, Dr. Nathan Freed, of course, for support, and Dr. Karen Chang, who assisted us with the coding, and Tom Skipper, who assisted us as well, and of course the NIH MARC program and Rutgers University Camden.
Questions?
There were no questions. I checked the whole list.
Good. Yeah, go ahead.
Are there any questions on the?
No, there aren't.
Good, like that.
I have just a quick question. Did you guys get to see if like there were any other than the spike, were there any other common mutations that happened through your samples?
We were so it's like really it's like 26,000 like 26 kilobases.
It's around 30,
Yeah.
30.
Yeah, so there were a lot, so we didn't look through. As soon as we found the location of the S gene, we were like, let's just focus on this one, trying to figure out how to interpret our results.
Yeah, given that we had like a lot of references that pinpointed this location, we thought it was, a safe bet to go look into this location because we were able to find what we were looking for. So that's why we decided to look for that specific location. But in future studies, and we can be able to look at different locations, see if like, oh, are there any other common mutations?
Yeah, but there weren't any papers that indicated that there would be similarities between mutations on the alpha and the delta. Literally, that one mutation, I think they had in common.
Thank you guys.
So same question. So what was the most challenging aspect of your training this week?
Well, definitely the coding. John and we took most of the coding responsibilities, so we were kind of a nice little team right there. So I would say that was definitely difficult, in my opinion, personally, trying to navigate errors and whatnot.
So I'd say after we did the coding and we received the results, I believe the analysis, the DNA sequence analysis, was a bit difficult to understand just because I haven't taken any genetic classes or this is something new to me, so that would be something that was challenging.
So we had to actually go through and search how to read this exact type of file to interpret it and actually understand our results. And that was difficult because there's not much written about it. There aren't many examples, or people don't share their examples of files of how they looked when we got them. They only share their refined, finished report. So that was another challenge that we experienced.
But despite these challenges, I think we had a really good time, good bonding time this week. And I think we all learned a lot, and not only about coding, but also about, SARS and how big this actually is, like looking at the chart, how crazy, like from one variant to another, all these mutations can arise. So